Well, thank you so much, Peter, and thank you for coming out. Friends, I hope that at the end of the session you will consider your evening well spent. I must confess I've been so kindly treated while I've been in Australia. I would never have suspected the kindness of your hearts after watching the way your cricket team demolishes your little Kiwi brethren, but I now know there's a warmth to Australians that is very powerful. So thank you so much. So I thought maybe just a little bit of my biography, how I, as a cell biologist involved in cancer research, became involved in the very controversial issue of evolution. Certainly when I started university, I had no interest in evolution. I thought it was sullied by controversy, and maybe it was just a quagmire of disagreements. And anyhow, I wasn't really interested in fossils and comparative anatomy and comparative behaviour. What I loved was DNA and proteins and cells. And so I was never going to think about evolution apart from the compulsory first year courses. Small things amuse small minds. I became a cell biologist. I cut off about 10 microns. That's my limit of interest. But over the years, as I got involved in cancer research, I stumbled across, really, new developments in the literature which linked cancer cells and living organisms together in an amazingly close way. Because the principles of genetics which were studied in cancer were exactly the same as the principles of genetics which are studied in evolution. And so I promise you I'm not an evolutionary biologist, but studying the evolution of tumours took me inexorably into reading and coming to a new understanding about the evolution of species. And so the book which Peter mentioned, in a way I'm amazed no one else has written it, because it was screaming to be written, but maybe I was just at the right time at the right place, fascinated by clones of cancer cells, and that led me into the study of clones of species, if you like. So what I'm going to do today, half the time I just want to talk about some of this astonishing evidence. I won't do it systematically, I'll tell you some stories. And then after that we'll think about what does this mean for Christian theology. And in particular, what does this mean for people who are committed to faith in the God of the Bible and in the Son who has revealed him, our Lord Jesus Christ. So that's where I come from, fully committed to mechanistic science, which I see as a small part of my faith in the living God who's been revealed through Jesus. So first of all, I want to tell you a little story. This isn't by no means a new one, which will just help me to explain some of the genetic approaches which I will be following. Now I want to talk about cheek muscles, believe it or not, which help us masticate our food. And I've just got a simple diagram here. I'm afraid I haven't taken the originals because I don't want anything which might be a problem for copyright. But we've got two sets of cheek muscles here. And on the left-hand side, this is a cross-section through the cheek muscles from the macaque, okay, which is an old-world monkey, and like all apes and monkeys apart from human, it's got very powerful cheek muscles, a very strong face, if you like, because it needs to tear its food apart or whatever. Monkeys have a very tough diet. So there are two sorts of fibres here. Again, this is a cross-section. The ones stained red are type 2 fibres, and they contain a muscle protein called myosin heavy chain 16, which is involved in giving power to muscles when we chew. And the small fibres are type 1 fibres, and they aren't particularly interesting to us. So type 2s and type 1s. But if you look at the human cheek muscles, our chewing muscles in cross-section, you see that our muscles, we as a species, you and me, our cheek muscles are wizened. They're small, they're atrophic, they're not at all appropriate for eating raw meat or nuts or whatever. And the difference is that the type 2 fibres are highly shrunken. Now, this is a feature of all of us. Our type 2 muscles are shrunken, and our cheek muscles, are much weaker than those of any other primate. Why should this be? Let's look at the genetics. So the next picture I'm going to show you will be a number of species looking at part of the myosin heavy chain 16 gene, which encodes for this muscle, which in other primates fills type 2 fibres. So here is a little section of genetic sequence. And we'll just think of, let's think of the woolly monkey for the moment down on the bottom. So the woolly monkey, it's a New World monkey, and I've put these letters in groups of 3 because our genetic sequence is read in 3s. So these letters, these A's and C's and T's and G's, they are the 4 chemical letters which comprise our DNA sequence. And one set of our genome has got 3 billion of these A's, C's, T's, and G's in all sorts of patterns and orders. And that directs the production of our body plan. It's our blueprint, if you like. So you can see I've got them in groups of 3 because these 3 letters, these 4 letters in our DNA direct the production of proteins, such as hair protein, if you've got any, and insulin and digestive enzymes, and muscle proteins like myosin H16. Now just look at these, and you'll find, going back to the human now, let's just read it through. ACC, well, we are the same as all these other species. We are really close, aren't we, in many ways? So humans, chimps, bonobos, gorillas, and orangutans. We are classified as great apes. Then there's the rhesus macaque and the pigtail macaque. They are old world monkeys. And then the woolly monkey is a new world monkey from South America. So we've all got this triplet here. So that's going to encode, in every case, the same amino acid, which makes a protein. And then there's a CTG, that's pretty similar. A CAT or a CAC in one case, pretty similar. AGC, very similar. And then I've boxed something which is remarkable. You see, where every other species starts the next triplet with an A and a C, I've left a blank. You can see that? A blank. And what that blank is a mutation. 2 bases, 2 of these chemical letters have fallen out of our DNA sometime in our history. And our muscle gene now lacks those 2 bases. And this is a rather devastating effect. It's going to screw up the gene and screw up the protein. And so the normal protein will not be produced. In fact, what I've done at the bottom is just to show what this effectively does when 2 bases fall out. So this is the human sequence up here, the same as the very top one, and the new human sequence with a new triplet phasing. So if you drop out 2 bases, then the new triplet becomes C, and then the GC from the next triplet. You can see it's a frame shift. And then you have the frame shift, the base, the triplets of 3 are out of phase from now on. So what does this mean? It means that we as humans are mutants. That's a characteristic of all of us. the myosin H16 gene is scrambled, we can't make that protein which all other primates make. And so we've got these delicate human features. Truly, some of us, it's gaunt, but generally it's a human-specific shape with small muscles. So we get by, even though we're mutants, even though our type 2 fibers in our cheek muscles lack this protein because of this crippling mutation. Now, why should this be? No one knows. There have been some suggestions. Perhaps some people have said this mutation was advantageous because by weakening the muscles in our jaws, it enabled our skulls to shape differently so they could come over our eyes and make room for more brain, the frontal cortex. The timing is probably wrong, so there's no good reason known, but the fact remains that what separates us in this gene from all our other primate friends and relations is a 2-base pair deletion. We humans are different from all other primates. Tested anyhow, and this is easily testable by just recruiting more primates and sequencing their DNA. We differ from all other primates by a crippling 2-base deletion. So somewhere in our history, an ancestor underwent that mutation, lost the power of their cheek muscles, and subsequently that mutation was spread to all the other members of the population and thence to all humans. And in fact, people have looked at different humans of different ethnicities, and regardless of where you come from, you have the same mutation. So it's a feature of humanness, and it separates us, it's a chemical change which separates us from other primates. So we don't know why it happened, but it did happen, because that's a very simple inference from simply looking at comparative gene sequences. Are there any questions about that at this stage? Yes.
Couldn't God have just made us like that?
Could God have made us like that? Of course he could have. But the point is, when you come across a natural event, the most obvious thing to do is to attribute it to the common chemical processes which are familiar to us. Because this sort of mutation is seen in a clinic, in a genetics clinic every day, every week. These are common mutations, thoroughly studied, inducible by different sorts of agents and whatever. And so it's not necessary to call upon divine action. Rather, we see these things as representing the secondary causes which God ordains and maintains. Yes.
Perhaps just on that one example, could you arguably say the reverse, which is the mutation led to the gene that the monkeys have?
No, because it's far easier to say that the mutation occurred in one species than to say that the identical mutation occurred in 300 other species. So that doesn't really work. But let's move on. Let's move on to another study. Sorry, maybe I should mainly keep questions for the end. Now, this is a picture of Gout. That's an artist from 1799 who experienced the horrors of gout, which is something, it's the king's disease, isn't it? Something that comes across certain people who live in a rich diet. The gout is another issue which reflects back on our genetics. So I'm going to show you now some more gene sequences which illustrate exactly why certain human beings suffer from gout. And again, I'll be lining up the human sequence of a particular gene with that of a number of other primate species. Now, in this case, we know that gout arises from crystals of uric acid, which is a breakdown product of DNA. So if we have DNA-rich food, we can get lots of uric acid into our system. And that uric acid can form little needles, and those little needles can be taken up by inflammatory cells in our joints, and the result is, ouch, redness, inflammation, pain, and heat. Tumor, rubor, K-law, and Dolor, for those of you who've studied inflammation. And here is the urate oxidase gene, and what we could call the pseudogene, present, here we go, in humans, chimps, gorillas, orangs, rhesus macaques, baboons, owl monkeys, and crab-eating monkeys. And you'll notice that with 4 species, there is one common mutation. A C down here, the crab-eating monkey and the owl monkey are New World monkeys from Latin America. The baboon and the rhesus macaque are Old World monkeys. And the orang, the gorilla, the chimp, and the human are, well, biologically we are apes. And here a CGA has been changed by a 1 base pair deletion in our DNA to a TGA. Now TGA doesn't encode for a part of a protein. Rather, it is a sign which says stop. Stop making the protein. It's the punctuation at the end of a protein chain. So this is another well-known, devastating mutation encountered daily in hospital genetics labs, where a triplet, which specifies an amino acid and a protein, has been changed to a stop. So at that point, the protein terminates. The protein, and it's not made anymore beyond that point, and a truncated protein tends not to work. So in 4 species, humans, chimps, gorillas, and orangs, we've got exactly the same mutation. Now, this comes back to one of the questions I was asked earlier. What happens when you have multiple mutations, or one mutation, shared by multiple species? The inference is the mutation occurred once, because these are very rare mutations. The mutation occurred once, and all the species which possess it now received it by inheritance. So the inference from this is that humans, chimps, gorillas, and orangs are all descended from an ancestor, in the reproductive cells of which a TGA mutation arose to scramble the gene, and then all the species which now contain that mutation are descendants of the single reproductive cell where that mutation occurs. And that seems very plausible because on all sorts of other grounds, we know that humans, chimps, gorillas, and orangutans are the great apes, biologically speaking. So there are other grounds, anatomical and whatever, for postulating that these groups form a natural category. And here is very strong evidence for a shared mutation. 4 species. The bonobo is not there, the pygmy chimp, but you can bet your biology it will have the same mutation. That's testable. 5 species, same mutation, descended from the common ancestor in which that mutation occurred. And that's why we get gout, because we don't have the urate oxidase gene, so we have levels of uric acid in our blood which are much, much higher than those of nearly all other mammals. Sometimes so high, it precipitates as needles and gout ensues. But let's move on. In fact, I'm going to need to hurry a bit, I think. Here's a nasty tick. I hope you don't have it in Australia. I think it's an American tick. And it bites people, and some people then become sensitized to red meat. After a meal of red meat or pork, kidney or something, these people can end up with an allergic reaction which can be anaphylactic. It can be life-threatening. Why is this? It's because most mammals have a certain sugar structure on their cells called alpha-gal. But we don't. And if this tick has bitten a mammal with alpha-gal, and then comes and bites a human being, and we don't have alpha-gal, then the human being can make antibodies against that alpha-gal containing protein that the tick injected into them. And then when you subsequently eat red meat, you're sensitized and you can have an anaphylactic shock. The question is, why don't we have alpha-gal? It's because as human beings we lack an enzyme called alpha-1,3-galactosyl transferase. We lack that enzyme. It's there in our DNA. We carry it in our DNA. It's there but non-functional. So we can't make alpha-gal, but when another bug injects alpha-gal into us, we respond to it as something foreign and make antibodies and become allergic to it. So just let's look here. Here again, comparative genomics. Human and orang, we're the great ape representatives, macaque, the old world monkey, the marmoset, and the capuchin. And I've just got 3 mutations here which are found in alpha-1,3-galactosyl transferase. And you can see here in exon 7, which is a coding sequence in the gene, a G is missing, right? The G's fallen out. I'll show you a detailed sequence later. So that's a crippling mutation. And it's shared by humans, orangs, and macaques. So we need to go a bit back further now and say here is a very infrequent, rare mutation. And it's shared by apes and old world monkeys. And the implication, therefore, is that apes and old world monkeys are descended from a common ancestor in which that particular mutation, that particular rare mutation occurred. It's just not possible to think of that mutation occurring independently in various species, especially as if you look a little further along the gene, you find a TAC or a TAT going to a TAG, which is another one of those stop mutations. And that's also shared by humans, orangs, and macaques. And then if you go a little further, you find another basis fallen out. That's another devastating mutation. This one is only shared by humans and orangutans. So by looking at which species share which mutations, you can put together a family tree. And here we've got a family tree of the primates, a very simple one. Humans and orangs are derived from the common ancestor in which this mutation occurred. Humans, orangs, and macaques are derived from the common ancestor in which those 2 mutations occurred. So here are 3 mutations which tell us something about our distant past. And they also tell us, they have implications for today, why certain people can have an anaphylactic shock after eating red meat. Here is just one of those mutations, just against a slightly larger segment of the gene. I've just crammed it all in now, just for the sake of space, against an increased number of species. And again, we can see here we've got a 1 base deletion only in humans, orangs, and macaques, not in marmosets or howler monkeys or capuchins, or in lemurs or lorises, which are also primates, but they're prosimians. So once again, shared mutation, a number of different species, indicates that when this rare mutation occurred, it was inherited by all the species which now possess it. Why did we lose that gene? No one knows. People speculate. To my knowledge, there's no clear answer as to why that gene became redundant, unnecessary, dispensable, disposable, why it was lost. But the fact is, it is lost. And it has big implications for red meat lovers if they get bitten by certain bugs. Interestingly, another source of danger for human beings is that some antibodies made in other species can have alpha-gal tagged on These are therapeutic antibodies. And some people given therapeutic antibodies in hospital can suddenly mount violent allergic reactions against alpha-gal, tagging antibodies made in other species. There's practical implications of our past catching up to us. I've been very negative. I've talked about genes we've lost. Here is a wonderful story about a gene we've gained. And, well, many of us have gained. The glory, the wonder of three-colour vision. There's a beautiful rose, and I know that a significant number of men especially lack three-colour vision. Most New World monkeys have two-colour vision, with one exception, but we need to go into that. But generally, New World monkeys, the more distant relatives in the primate family, have two-colour vision. But Old World monkeys and apes have three-colour vision. How did that happen? And here this first picture just gives you a broad outline. On our X chromosome, remember, colour vision is linked. It's sex linked, so it's on the X chromosome. There is one of the opsin genes which encodes a protein which is responsible for colour vision. And it's next to another gene called TEX28, which you don't need to worry about. There is also another opsin gene. So with New World monkeys, there are two opsin genes, one on another chromosomes and this on the X chromosome. Way back in history, a block of DNA about 36,000 bases long was duplicated. And such duplications are known to occur in genetic systems, often through errors of DNA replication when DNA is made. So you can see I've just given you an idea of how that might be. Here's the red box, which indicates those 36,000 base pairs, which includes part of the TEX28 gene and all of the opsin gene. And that vast spread of DNA, 36,000 base pairs long, was copied and then pasted back into the DNA. So copied and pasted. So it was effectively duplicated. Again, these are well-recognized genetic events. Mechanisms pretty well understood. So again, answering your question, you don't have to postulate divine action for something which has a chemical explanation. So the outcome of that was one TEX28 gene, one opsin gene, a truncated TEX28 gene, which doesn't make a protein anymore because it's only a fragment, and a new opsin gene. This stands for long wave, sort of reddish, yellowish, and this is medium wave, greenish. So it's that duplication of a gene on a 36,000 base pair segment which gave us the wonder of three-colour vision. And we'd be sure of that. That's the royal we. This isn't my work, of course, because we can look at the DNA sequence and we can see exactly what happened. Now, when this segment was copied and pasted, you can see that there was a discontinuity at one end. At the other end, it slipped in seamlessly into the appropriate sequence. So, if we just look at the left-hand side of this whole complex, in humans, and also these two genes in New World monkeys, that's the sequence. And you can see that we humans and New World monkeys still have pretty much the same sequence to the left-hand side of this opsin gene. Only for us, it's a long wave opsin. We've got two here, whereas the New World monkeys only have one. But if you look at the other edge of the DNA duplication, you find a sudden breakpoint. Here you can see the AAT is exactly the same. This is the same segment, it's just been duplicated. TTGG, it's all the same, and suddenly at this point, it's different. Because we've moved from the left-hand end of the opsin gene to the right-hand broken end of the TEX28 gene. And and the breakpoint is exactly the same in every case. This was a single event, a single remotely probable event, and it's shared by humans, chimps, and Diana monkeys, and obviously any other old world monkey. You can look in that and you'd expect to find exactly the same breakpoint. So here was a huge mutation event which occurred in an ancestor of apes and old world monkeys, which gave us a new colour gene so that we could see three-colour vision. Sadly, the reverse often happens. Now you've got this array of genes with a lot of duplication. You can often have one lost and you revert to two-colour vision. So human colour blindness is simply reverting to the New World monkey conformation at this locus. But here we have a mechanism clearly understood, clearly unique mechanism shared by humans and other apes and old world monkeys, arose once, demonstrating not only the origin of a new gene and a new physiological capacity, a sensory capacity, but demonstrating a common ancestor because it's the same mutation. Here's another amazing story where we have gained a capacity. What am I going to talk about? The placenta. Okay, the placenta is, in evolutionary terms, a new organ. Only mammals have a placenta. And you can ask the question, where did the placenta come from? Well, obviously it was a multi-step process. But the cells on the outside of the placenta, again, they vary in different mammalian orders, but in higher primates, they form what's called a syncytial trophoblast. Here are trophoblastic cells. Sorry, this is a cell biologist cell with a membrane and a nucleus in the middle, and that's a cytoplasm. That's all we need to know, individual cells. But as the placenta forms, so that the baby's blood supply can invade the mother's uterus and receive receive the nutrients and oxygen from the mother, the outside layer of the placenta fuses. The different individual cells fuse to form enormous giant cells called syncytia. So the outside layer of the placenta is called a syncytial trophoblast because it's syncytial. A ginormous cell, continuous cell, with the nuclei of all the progenitor cells inside it. One reason for this, perhaps, is that a syncytial trophoblast doesn't have cracks. You can see individual cells might have cracks that immune cells can squeeze through. But a single cell, a continuous cell, a syncytium, doesn't have cracks, doesn't have joins, so immune cells from the mother can't penetrate the fetus and recognize paternal antigens and cause damage. So if you've ever wondered why a mother's immune system doesn't attack a baby because of the father's proteins, here's one reason. In this case, we know how the syncytial trophoblast arose in higher primates. And it arose because a reproductive cell was infected by a retrovirus. Now we know about retroviruses. This is where my background as a cancer biologist comes from. Some retroviruses cause cancer. Human T-cell leukemia virus type 1 causes cancer in some parts of Japan, Africa, southwestern United States, southeastern United States, and other places. So these are potentially pathogenic viruses. But what these viruses do, they have a little tiny genome of about 10,000 bases, these A's, C's, T's, and G's. And they slip that when they infect a cell somewhere into the DNA of the infected cell. And here is where one of these viruses has gone. It's called endogenous retrovirus, endogenous meaning it's part of our DNA, so we all inherit it. WE1, and of particular interest to us is the ENV gene. Now, one interesting thing when an endogenous virus comes in is that it selects a little piece of DNA of the host cell called the target site, and it duplicates it. And then the virus slips its DNA between those bits of the host cell. And where I've got WE1, that's actually 10,000 C's, T's, G's, and A's. So there's a huge expansive virus in there. And you can see that here, this particular virus is present in exactly the same place in humans, chimps, gorillas, orangs, and lesser apes, gibbons. What does that mean? The genome is such a big place, the virus has got lots of options. You never find two viruses going into the same place because our genomes are so big. What this means is that in a single cell, this arose and got carried subsequently, so that all species which have inherited this endogenous retrovirus received it from the original reproductive cell where that mutation occurred. Then if you look at a couple of New World monkey species and prosimian species, and in this case even the dog, you can see a very changed, but never mind, the same sequence. There is the undisturbed target sites. So again, to a biologist, this is proof of common ancestry. I know if you're a philosopher, you'll laugh at me because there's no such thing as proof, but to a biologist, somebody working in a hospital lab, this is proof of common ancestry. If you see a tumour with a virus in the same part of every cell, you know that every cell is descended from the single cell in which that virus arose. Now, the relevance of this to the placenta, this retrovirus makes an ENV gene, and that normally enables the virus to stick to cells so that it can invade those cells. But now it's part of our DNA. It enables cells to stick to each other so that they can fuse and become a syncytium, form the syncytia trophoblast, which is essential in the life cycle of every one of us. So we as human beings are alive only because of this particular endogenous retrovirus, which makes a protein which was once part of the virus infective apparatus, but now enables cells to stick together in the placenta, where the syncytia trophoblast can form, which is an integral part of our development. I think I'll just pass over that one. I'm running out of time. Look, I'll finish with something that came out last week. If you've read my book, you've probably come across the other stories, but this is one hot off the press. And as Peter said, I did my PhD many moons ago in cellular immunology, really when it was still witchcraft, before it was a science. But I've never lost my wonder at immunology, because the cells which comprise our immune system make a potentially infinite number of antibodies. Okay? That's the whole basis of immunization. You can be immunized against something that you've never encountered before. And hey presto, your body makes antibodies against them and protects you from further exposures. So we can potentially make a zillion antibodies, but we've only got 3 genes for antibodies. 3 genes for antibodies, but the antibody proteins we can make a practically infinite number. How can this be? Have you ever thought about that? Maybe you haven't realized that there's only 3 antibody genes, but that's all we've got. The thing is, as a cell develops, these are called B cells. They are the cells which are responsible for making antibodies. So a pre-B cell, an immature cell in your bone marrow, it's got those 3 antibody genes. But as every B cell develops, the antibody genes are to some extent randomized. There's a procedure which causes directed mutations. So these are unspecified, unpredictable, they are random within limits. And so that every B cell which develops in our bone marrow ends up with a new, unique antibody-forming gene, which makes a unique antibody. So it doesn't matter what you're infected with, you will have some B cells circulating in your blood, which will enable you to make antibodies, whether it's a virus or a bug from Pluto or a chemical compound which is tagged to another protein or whatever. How does this happen? Well, it's been assumed, this is the process. So for the main antibody gene, there are different segments, V, D, J, and constant. And proteins called RAG1 and RAG2, which cause one of these to join to one of these, to join to one of these. So that you can sort of multiply the possibilities. So this gives you hundreds of possibilities to give you one particular VDJ segment, where one V has been joined to one D and one J. But the thing is, the joining mechanism is also sloppy. So the joints also vary in the DNA. And that means that this is where you have an infinite number of possibilities. Because the joining mechanism is somewhat random, you can make an infinite number of antibodies. It's the most amazing thing, studying immunology. The fascinating thing, people for years have looked at these proteins in our cells, which assemble the genes, and say, these look very much like little parasites called transposons. These are little segments of DNA which live a pretty independent life in different bodies and different genomes and jump around. They copy and paste and potentially do mischief, but generally they're innocuous. They said, these proteins look as if they come from a transposon. We don't have a transposon that looks like that, jumping around in our genomes. Until last week. And a transposon which jumps around, which looks very similar, was discovered in Amphioxus, the lancelet, the most simple of all vertebrates. If it is in fact a vertebrate, hang on, my biology fails me for a moment. Anyhow, I think it's the most simple of the vertebrates. And here is a little segment of one of the RAG proteins in the lancelet. And here in bold, I've drawn, this is now looking at the amino acid sequences which are very similar or identical in our RAG1 genes, which are active in immune cells, to the lancelet, or a similar sequence which is found in the CH. Look, here's a fascinating story. Here are two bases which have fallen out. Whether you're a human, a mouse, a platypus, a chicken, a frog, a zebrafish, or a shark, you've got an immune system, okay? You've got RAG proteins, which are symbol antibody genes, but you've got two amino acids missing, which are present in the presumptive progenitor. The implication is that, yes, something like this lancet jumping gene 550 million years ago was the progenitor of our RAG1 gene, which is responsible for producing our immune system. And maybe two amino acids had to drop out, 6 bases, in order to convert the transposon gene into the immune gene, which all of us have. And in fact, if we don't have it, we'd have to live in a bubble because we would be sensitive to every bug which came along. So that's just a too long skim, over some biology. But what does this mean for us who are Christians? And I'd like just to quote a book I read quite recently. on Origins by an American theologian. He says, rightly approached, science will become a benefactor to the faith of our time. If you feel threatened, there's no need, because as we discover truth, it can only enlighten and illuminate Christian faith. Rolnick says, a careful contemplation of scientific discovery can bring us to a broader and deeper love of creation. I would phrase that slightly differently, perhaps a broader and deeper love of our Creator. Evolutionary theory is a potential gift for theology. Honestly confronted, evolution causes a shift in how we think about creation, not a loss of faith. So, if we have time, and I know I must shut up, let's just think about 4 areas where I think as Christians we can be enriched by these studies. Now, the first thing is reading Genesis. And lots of people read Genesis in such a way as to find that these evolutionary insights are incompatible with their understanding, certainly, of Genesis 1 to 3. Let me quote from Barton and Wilkinson, and I think Wilkinson was mentioned a little earlier, I think, in conversation. And this book says you cannot put good questions and expect fruitful answers from a text apart from a grasp of the kind of material it is in the first place. Misjudge the genre, and you may skew any of the things you try to do with the text. You see, friends, I truly believe that although science should not affect the way we read our Bibles, if we import wrong presuppositions into the way we read our Bibles, then science can act as a corrective. Science can alert us to hidden presuppositions which are directing us perhaps in the wrong way as we read ancient texts. So yes, science can help. Genesis is derived from a genre of Near Eastern creation myths. So that's a proposal from the same book, biblical scholar Louth and Barton and Wilkinson. So genre is so important. I'll just give you a couple of examples and then we'll go back to Genesis 1 to 3. Those of you who are old like me may well remember in the premier scientific journal about 15 years ago, on the April the 1st issue, an article by a very famous scientist called Robin Weiss called On the Dorian Grey Mouse. And those of us who maybe were a little aware of the date read this article and didn't read quite straight. It was a bit funny, in fact. It was an April Fool's Day joke in Nature magazine. Does anybody remember that, the Dorian Grey Mouse? The feedback came a few issues later. People who weren't in the Anglo-Saxon tradition, some of them were highly irate because they didn't know about April the 1st. They didn't read it as a spoof. They read it as a rather surprising scientific article. And a group from Spain said, look, we stopped what we were doing. We started putting it into grants, applications for funding. It's only later we found that it was a joke. And we all sat down and had a good laugh. It is a joke, isn't it? Somebody from Italy said, this is highly irresponsible. When science is struggling with credibility in the media, what do you mean you're doing by something like this? Folks, the issue is genre is critical. Read it as a spoof, you get a good laugh. Read it, in this case, as a scientific report, you are up the creek. The second example is the Gospels. You know, for much of the last century, in universities, most Gospel studies have been done on the paradigm of Rudolf Bultmann, who argued that the Gospels are really just a pastiche of early church stories put together like beads on a string, reflecting controversies within the early church. And that's what most theology students are taught. And then in the 1990s, somebody set out to confirm this, called Richard Burridge. And he looked at the genre of the Gospels in comparison to the genre of of biographies from the ancient world, looking at historians such as Tacitus and Suetonius and others like that. And it totally convinced him, surprise, surprise, that the Gospels in fact belonged to the genre of bios. They were in fact biographies. They were in fact about Jesus. It seems rather obvious. But Richard Burridge broke out of the Bultmannian paradigm, which had totally misrepresented the gospel, and generations of New Testament students, and found that the Gospels are biographies in the ancient sense of the term, of Jesus. They are occupied with telling people who Jesus is. It's a wonderful book. It's a rather technical book, but he's just comparing various features of biography with the Gospels and finding that the Gospels sit firmly into the known genre of biography. So it's very enlightening. Folks, the Gospels are about Jesus, if you ever doubt it. So Genesis, then, is an ancient Near Eastern creation story, and there are so many pointers to that. Like many of the ancient Near Eastern creation stories, they start off in a garden paradise. Adam, or to the Babylonians, Adapa, really means, it's linked to the word earth. He's an earthling. We are earthlings. Made from dust or clay, the stuff you walk on. Dust in the Bible, clay if you're a Babylonian. Put there to cultivate the land. In these stories there is a plant which gives immortality. The acquisition of wisdom means becoming like God, Yahweh, or the gods. There's a serpent, there's nakedness, there's lost immortality, there's a flood story. Current historians, archaeologists, Old Testament scholars recognize that the early chapters of Genesis belong to a well-known genre. It's the genre of the ancient Near Eastern creation story. But it's very distinct. The form is the same, the content is radical. And here, instead of all the horrible, feuding, sadistic, bloodthirsty gods of Babylon and Egypt and Mesopotamia, we have here a God of supreme authority. The sun and the moon and the stars are his creation. Set in the sky as lights. It's so significant. I'm sure probably all of you have heard this before. Hebrew had very good names for the sun and the moon, but they aren't used in Genesis 1 so as not to dignify them with appropriate name. They're just lights. Just things which Yahweh has made. And the sea monsters which represent the incredibly terrifying forces of chaos to the Babylonians. These are creations of Israel's God. This story speaks of the sheer goodness of creation. This story introduces humanity in a new way, from the breath of God, not from the blood of God. There's nothing divine in Adam. We are from the breath of God. We are God's image. We aren't just a slave or an afterthought, as the Babylonians might have thought. We're God's image. Only the king was the image to the Babylonians. And unlike the Babylonian stories, humankind is sinful. So it's a familiar genre with a blindingly radical message. And it just fills me with wonder that little tiny Israel, little struggling Israel, should have such a sublime vision of her God when she was surrounded by these huge, powerful, cruel, imperialistic, militaristic nations who lived under the fear of ever-warring, sadistic, horrible gods. Here is Israel, which somehow had the vision of a God of love and holiness and truth. There's the moon God. My former theology professor, Genesis 1 is a mockery of the gods. a radical de-divinization of creation. And for a scientific audience, how important was that for the development of science? Science could only arise when nature was de-divinized, when the spirits were taken out of the rivers and the trees and the animals and the elements. Nature was de-divinized because there was only one God. We were free to look at the phenomena around us. So this is a polemic against the surrounding religion. Isn't it amazing? Little tiny Israel doing that to Assyria and Babylonia and Egypt. How did Israel come across such an amazing vision were it not somehow revealed by that God approaching humanity and causing humanity to itself? So, dear friends, how should we think about Genesis? You know, we spend all our time agonizing about whether Adam was a Neolithic farmer and when he lived and how to relate the fall story with physical anthropology and all this sort of thing. Can I suggest, if we're going to be true to the genre, which is Genesis 1, for Christians anyhow, we should be developing models of how followers of God, followers of the God of Jesus, should be living in a neo-pagan society. That is the model we need to develop from Genesis 1. A society which is now ruled back by some of the old gods, such as Hedone and Narcissus and Mammon and Asherah, the gods of sex, the gods of power, the gods of money, the gods of sheer selfish enjoyment. Folks, to me, this is the message of Genesis 1 if we're going to be faithful to the genre. We need to go back to the loving and holy God of Israel. And here we will find the remedy for the ailments of our society. I was just listening to our pastor speaking in church last week, speaking about Asherah and how Israel became contaminated by this importation of the foreign god of reproduction and sexuality and fertility. Sex has become the highest, the summum bonum of our pagan society, hasn't it? And just walking down the street afterwards, there was a New Zealand newspaper saying, speaking of the loneliness of modern life, separate sex and relationship, and what have you got? Broken people. I think Genesis 1 should take us back to the healing, restoring, life-giving God that the ancient Hebrews worshipped. Very briefly then, just 2 or 3 more points. Creation. I believe that as we understand evolution and put it alongside our Christian faith, we can come to a new understanding of the meaning of creation. This is just a lovely NASA image. NASA lets you download their images so long as you give credit. So there are light echoes used to study protoplanetary disks. Please keep your questions for Lewis. Creation is the gift of being. Right, if I may quote Professor Polkinghorne, creation is a doctrine of ontological origin, not a concept of temporal origin. Creation means that God gives existence to all things at all times, not that he does certain things at certain times, as if he has a 6-day week and he's a trade unionist. I think this is one thing that seeing evolution, seeing it as a part of creation, it sets us free. Not to say, do you believe in creation and evolution? But to go back and define creation biblically. Creation biblically is the gift of being at all times from the God and Father of our Lord Jesus Christ. Whatever that being might be, whether it's one universe or many or infinite, whether it's our universe or the quantum vacuum, whatever physical reality you can conceive of, that belongs to the circle of creation. That is given its being by the God Israel. Within that creation, you can then study histories. The big red line from the dot in the middle to the edge, cosmic history perhaps. Okay, so there's a role for astronomers here. Cosmic history is not an alternative to creation. It's part of it. The blue line, the little green line at the bottom, evolution is history. It's part of God's creation. And therefore it cannot be held as, in any sense, opposing creation. It is just one of the histories of creation. So we can think of the category of creation. There's physical reality, humanity, Israel, the church, each individual. These are things which God creates. Or human beings can share in that. Maybe this is part of our bearing God's image. Art, sculpture, music, architecture, even raising our children. That is creative. If you bash your children up every day, they'll end up violent. If you pour your love into them, then hopefully they will pour their love back into other people. That's the category of creation. The category of history. Cosmic, terrestrial, biological, primate, civilization, Roman Empire, Baroque music, the SPCA, the Labour Movement, university history, whatever. These are histories. So I marvel at evolution because it tells me about history. It is the history within the creation. Okay? Walk around town doing this. Just remind yourself, creation is an absolute concept. History is of all sorts, whether you do it in an arts faculty or a cosmology faculty. They're the atoms, which are part of this. Maybe I'll be missing this because I don't know. Just finish with another point. Does evolution mean we're just atoms? Just atoms. I'm sure we've all heard people saying, well, I didn't come from a monkey. And I have this little game with my wife sometimes. She's a very wonderful musician, and she sometimes listens patiently while I blat on about science. We have this little game, well, gee, are we vertebrates? Oh yes, we're vertebrates. Are we mammals? Of course we're mammals. Are we eutherian mammals? Oh yes, we're eutherian mammals. Are we primates? Yes, we're primates. Are we anthropoid primates? Well, maybe. Are we apes? Oh no, we aren't apes. That's a term of abuse. So, where are we? Are we just apes? Does this just mean we're nothing but apes? Does this take away our human dignity? But again, folks, I think as I close, that this has brought to my mind sharp contrast between our genetics and our past and what it takes to make us human. And folks, so let me say, genes are not sufficient to make us human. Genes give us the potential to be human, but it's terrifying. Genes do not make us human. Genes, human genes, the genes of Mozart or of Lloyd George or anybody else, genes do not vouchsafe the survival of human society. What makes us human is personal knowledge. And so we have this very clear distinction we have to remember. Genes are necessary but not sufficient for the development of a human person. There's an interesting article that came out in Science a couple of years ago. Most, possibly all, human neurocognitive skills are shaped by culture, and many are culturally inherited. But the parallels between mind reading and print reading are extraordinary. Now, mind reading, I'm not talking about some freaky sort of telepathy. I'm talking about a basic human capacity to see in you and in you a human being like me. This is called mentalization or theory of mind. We become fully human only as we grow in theory of mind and mentalization. Genetics can't do that. Genetics can't give us theory of mind. We learn theory of mind on our mother's knees, and then in our wider family with our fathers or whoever brings us up. And then schoolmates and teachers, and so it goes on. We become human only in relationship. And so I've just got a few papers, I need not spend time in them. Here we are, interactive communication. Teaches us language, obviously. Theory of mind. Gives us the ability for mental representations of time, motion, space, pitch, colour, attention, learning, and memory. This is what we learn from people. It is not genetically encoded. You can find that in a journal called Cognition, 2015. Let's look at a few other things. Even more physical. I started off with a picture of the brain. Here's another one. Downloadable from the National Cancer Institute. Again, thanks to the Americans, they're very generous. You can use their government websites for free downloading. They just ask you to apply. So there are human brain trains. Folks, our white matter development depends on knowing. You imagine if, regardless of what happens to you, you still develop a liver and a kidney and lungs. But we don't develop a brain apart from human love and care and interaction. Here is a little 13-year-old boy from one of the Romanian orphanages. Neglect affects the way our brain is structured. Hippocampus decreases in size, the amygdala increases, there are changes in the anterior cingulate cortex. My son's working in the Centre for Brain Research in Auckland. I don't know these words, but suffice to say, it's knowledge, it's love, which affects the stuff between our ears in development. Horrendous thought. It's not genetic. Genetics just gives us the capacity to develop a human brain. Head circumference is reduced in children raised in neglectful conditions. And going back to this first article, if a group of human infants managed to survive on a desert island in a cruel Lord of the Flies-like experiment, they would be no more likely to develop a theory of mind and become explicit mind readers than to develop a writing system and become literate print readers. Folks, we become human only because of those who were human before us. It's relationship which allows our genetic potential to be expressed. Human love and compassion makes us human. It forms the stuff in our heads. Fascinating story. Have you come across Frederick II of Hohenstaufen? He is perhaps the one man in history who did the unmentionable experiment. He wanted to find out what language children would learn if they were taken away from their parents and never spoken to. Let's read what Friar Salambini has said. This is with Frederick II, 13th century, very cruel man, and he had lots of disagreements with the Jews. He bade foster mothers and nurses to suckle and bathe and watch the children, but in no ways to prattle or speak with them. For he would have learned whether they would speak the Hebrew language, which had been the first, or Greek, or Asia, or Latin, or Arabic, or perchance the tongue of their parents of whom they had been born. But he laboured in vain, for the children could not live without clappings of the hands and gestures and gladness of countenance and blandishments. It seems this horrible experiment ended in failure. Without being spoken to, the children died. They did not develop in the absence of interpersonal love. When I did my BTH, I did my correspondence with the University of South Africa, and I learned this saying. Any South Africans here? This is an amazing saying coming from Black tribes. It's got so much more wisdom than you will ever find in a lot of Western academics. Umuntu ngabantu means a person becomes a person through persons. And I finish with a quote of Jesus. Because as I think about the importance of personal knowing, in order for us to develop our full humanity, personal knowing, not genetics, personal knowing, will enable us to develop our humanity. Jesus in his high priestly prayer in John 17, praying for his disciples, he said, eternal life is knowing you, the only true God, and knowing Jesus Christ, whom you have sent. And I used to wonder how such a tangible thing as life could come from such an intangible thing as love. And now as we think what it takes to make us a human being, our humanity comes from knowing other human beings. And in that sense, when we are brought into relationship with the living God, I must believe that our humanity changes again. And we become partakers, sharers in the life of God himself. What the scriptures talk of as eternal life. Jesus said, no one knows the Father except the Son, and no one knows the Son except the Father, and those to whom he will reveal. I wonder what's going to happen to civilizations. People withdraw from each other for all sorts of reasons, whether it's the primacy of sex or the primacy of the electronic media. And I believe that it's in knowing each other and supremely in knowing the God. Yes, knowing him personally, knowing his love and his compassion and his forgiveness, that we become full persons. Thank you.